Between the year 1500 and the year 1700, an estimated 2 million sailors died at sea. They did not perish from cannon fire. They were not claimed by sudden hurricanes, or shattered against hidden reefs, or killed by pirates. They died because the very connective tissue holding their bodies together simply began to dissolve. It was a disease of terrifying, almost supernatural decay. Teeth would fall out from spongy, blackened gums. Healed wounds, scars that sailors had carried for decades, would spontaneously tear themselves open, as if the original injury had just occurred. Cartilage gave way. Blood vessels leaked under the skin, forming dark, bruised patches. This was scurvy, and for centuries it was the accepted, unavoidable cost of doing business on the open ocean. Ship captains and naval boards simply factored a massive mortality rate into their ledgers. On a long voyage, you expected to lose half your crew to this rotting disease. Half! Part 1. The Scourge of the Sea. To understand the magnitude of what happened on a single cramped ship deck in 1747, we first have to understand the sheer, unyielding terror of scurvy during the Age of Discovery. We look back now with the benefit of modern biology, and we know exactly what causes this disease. It is a severe deficiency of vitamin C, or ascorbic acid. The human body, unlike most other animals, cannot synthesize its own vitamin C. We have to consume it. Vitamin C is essential for the production of collagen, the structural protein that acts as the scaffolding for our skin, blood vessels, bones, and tissues. When you go months without it, that scaffolding literally crumbles. Your body forgets how to hold itself together. But in the 18th century, the concept of a vitamin did not exist. In fact, vitamin C would not be officially discovered and isolated until the 1920s, when Hungarian biochemist Albert Szent-Györgyi identified it, eventually earning a Nobel Prize for his work. Back in the 1700s, the prevailing medical theories of the day were based on tradition, superstition, and a lingering adherence to the ancient idea of bodily humors. Physicians believed illnesses were caused by an imbalance of fluids, or by foul air, miasmas, or by internal putrefaction. So when sailors began to rot from the inside out after a month at sea, the medical establishment offered a wild array of guesses. Some said scurvy was caused by eating too much salted meat. Others blamed the damp, cold air of the ship's hold. Some even thought it was a form of severe melancholy or homesickness. The crisis reached a breaking point for the British Royal Navy in the 1740s. In 1740, Commodore George Anson set out on a mission to circumnavigate the globe and harass Spanish shipping. He left England with 6 ships and nearly 1,000 men. By the time he reached the Juan Fernandez Islands in the Pacific, over 600 of his sailors were dead. The vast majority of them died not in combat, but from scurvy. Entire ships were left drifting, helpless, because there were not enough healthy men to work the rigging. The accounts of Anson's voyage were published and read widely in Britain, causing a mixture of horror and national embarrassment. It was clear that if the British Empire wanted to project power across the globe, it had to solve the problem of scurvy. Enter James Lind. Lind was a Scottish physician, born in Edinburgh in 1716. He began his medical career as an apprentice and eventually found himself serving as a ship's surgeon in the Royal Navy. In 1747, Lind was stationed aboard the HMS Salisbury, a fourth-rate ship of the line, patrolling the English Channel and the Bay of Biscay. The Salisbury had been at sea for just a couple of months, but that was long enough. Scurvy began to creep through the lower decks. Lind was intimately familiar with the disease. He had seen the putrid gums, the lassitude, the sudden hemorrhages. But unlike the physicians sitting in comfortable armchairs in London, Lind decided to do something unprecedented. He decided to test the prevailing cures in a systematic way. Part 2. The Salisbury Experiment. On May 20, 1747, James Lind initiated what is now widely recognized as the first controlled clinical trial in the history of medicine. The elegance of his experiment lies in its strict parameters. Lind understood, perhaps intuitively, that to find a true cure, he had to isolate the variables. He went down into the cramped, dark forehold of the HMS Salisbury and selected 12 sailors who were all suffering from scurvy. He was careful to ensure that these 12 men were in the same stage of the disease. In his own notes, he recorded that their cases were as similar as he could have them. They all exhibited the classic symptoms: the putrid gums, the dark spots, the profound weakness in the knees. Lind then grouped these 12 men into 6 pairs. He kept them in the same quarters, ensuring they were exposed to the same damp air and the same cramped conditions. Most importantly, he placed all 12 men on the exact same basic diet. They received water gruel sweetened with sugar in the morning, fresh mutton broth for dinner, light puddings, and boiled biscuit. By keeping the environment and the baseline diet identical, Lind established a control mechanism. Any difference in their recovery, he reasoned, would have to be the result of the specific treatments he was about to administer. He then assigned each of the 6 pairs a different daily remedy, drawn from the most popular medical theories of the time. Pair number 1 received a quart of cider a day. Pair number 2 was given 25 drops of elixir of vitriol, which is essentially diluted sulfuric acid, 3 times a day. Pair number 3 was prescribed 2 spoonfuls of vinegar 3 times a day. Pair number 4 had to drink half a pint of seawater daily. Pair number 5 received a potent, spicy medicinal paste made of garlic, mustard seed, and horseradish. And finally, pair number 6 was given 2 oranges and 1 lemon every day. For 6 days, Lind meticulously observed the 12 men. He recorded their symptoms, their energy levels, the state of their gums and their skin. The results were not just clear, they were staggering. The 2 sailors taking sulfuric acid showed no real improvement. The seawater drinkers remained gravely ill. The vinegar and the garlic paste did nothing. The men drinking cider showed a very slight improvement, perhaps because the cider contained trace amounts of nutrients, but they were still sick. But the 6th pair, the 2 sailors given the daily ration of oranges and lemons, experienced a near miraculous recovery. Within just 6 days, the transformation was so complete that one of the men was actually deemed fit enough to return to active duty on the ship. The other had recovered so well that Lind assigned him to act as a nurse to care for the remaining 10 sick sailors. In less than a week, Lind had empirical, undeniable proof that citrus fruits cured scurvy. He had defeated the greatest killer of the maritime age using a systematic, comparative trial. Now, the logical continuation of this story seems obvious. Lind discovers the cure, he rushes back to London, the Admiralty orders ships to stock lemons, and scurvy is banished forever. But the history of science is rarely that clean, and the human mind is stubbornly resistant to new paradigms. Lind retired from active sea service shortly after his experiment and returned to Edinburgh to earn his medical degree. In 1753, 6 years after the trial on the Salisbury, he published his masterwork, a massive volume titled A Treatise of the Scurvy. You might expect the clinical trial to be the centerpiece of this book, but it was not. In a book spanning over 400 pages, the description of his groundbreaking experiment on those 12 sailors occupied just 4 pages. It was buried in the text, presented not as a world-changing revelation, but as just another observation among men. Why did Lind downplay his own discovery? Because Lind, despite his brilliance in designing the trial, was still a product of his time. He did not understand the biological mechanism of what he had witnessed, the concept of a nutritional deficiency disease, the idea that you could get sick simply because a specific invisible compound was missing from your diet was entirely alien to 18th century medicine. Lind still believed that scurvy was caused by blocked sweat glands, damp air, and the accumulation of toxic humors in the body. He thought that the acid in the citrus fruits was simply acting to clear out the body's putrefaction. He viewed oranges and lemons not as a required nutritional baseline, but as an emergency medicinal intervention. Furthermore, citrus fruits were expensive and difficult to preserve on long voyages. You couldn't just keep crates of fresh oranges in a humid ship's hold for 6 months. They would rot. To solve this practical problem, Lind proposed boiling the citrus juice down into a thick, concentrated syrup, which he called a rob. The Admiralty, looking for a logistical solution, eventually tested this boiled extract. But here is the tragic irony. Vitamin C is highly sensitive to heat. By boiling the juice for hours to create the syrup, Lind inadvertently destroyed the very compound that cured the disease. When naval captains tested the rob on subsequent voyages, it did not work. The results were inconsistent, which only fueled the skepticism of the medical establishment. For decades after Lind's treatise, sailors continued to die by the thousands. Influential physicians in London, men who had never set foot on a ship, dismissed Lind's findings. They preferred their own theoretical remedies. They advocated for malt, for sauerkraut, for better ventilation in the ships. Some even pushed for the continued use of sulfuric acid. The bureaucratic inertia of the Royal Navy, combined with the academic arrogance of the medical elite, meant that the greatest medical discovery of the century sat ignored on a bookshelf. Part 4. The Legacy of Evidence. It was not until 1795, a full 48 years after the experiment on the HMS Salisbury, that the British Royal Navy finally made citrus juice a compulsory part of a sailor's daily ration. This policy shift was championed by a younger generation of physicians who had read Lind's work and had seen the undeniable proof of citrus on long voyages to India. James Lind died in 1794, just 1 year before the Navy officially adopted the very policy he had uncovered. He never lived to see scurvy eradicated from the British fleets. But the impact of Lind's work extends far beyond maritime history. What happened on the cramped deck of the HMS Salisbury in May of 1747 was a fundamental shift in human epistemology. Before Lind, medical treatments were largely based on authority, on ancient texts, and on unverified anecdotes. If a famous physician said that scurvy was caused by bad air, that became the accepted truth. Lind challenged that system. He asked a question, controlled the variables, applied different interventions, and let the results speak for themselves. He laid the groundwork for the randomized controlled trial, the gold standard of modern clinical research. Today, every new pharmaceutical drug, every vaccine, every medical device must go through rigorous controlled testing to prove its efficacy and its safety. The methodology we rely on to protect human health across the globe traces its lineage directly back to a Scottish surgeon rationing out cider, vinegar, and lemons to 12 exhausted men in the Bay of Biscay. Lind's story is a powerful reminder of how difficult it is to see the truth, even when it is right in front of us. He built the perfect experiment, captured the correct data, but was blinded by the medical dogma of his era. Yet the methodology he pioneered survived. The framework of the controlled trial is built to strip away human bias, to filter out our preconceived notions, and to force us to confront what actually works. It took nearly half a century for the establishment to catch up to the data, but the data ultimately won. Millions of lives were saved not by a sudden flash of divine inspiration, but by the careful, systematic observation of 12 men in the dark belly of a wooden ship.